So my name is Parren, and I'm Head of R&D and Executive Vice President at Therapeutics. Today I'll talk about a bispecific gamma-delta T-cell engager platform that we're using to develop novel treatments for cancer. Now the antibody landscape is blooming. Earlier this year, the FDA approved the 100 monoclonal antibody product. And this was reviewed by Ashley Mullard in this commentary in Nature Reviews Drug Discovery. At this time, we are already at over 110 approved and marketed antibody products in the EU and US. And also, at the same time, more than 950 antibodies are in clinical development. Now, looking at the various antibody formats in development, this landscape is shifting gears. So we're moving from canonical antibodies, so these are IgG1, human IgG1 antibodies with regular architecture, as pointed out here on the left of the slide. We're moving to antibodies with increased potency. So these are formats that include antibody-drug conjugates, as well as bispecifics. So bispecifics are antibodies that have the ability to bind to different antigens. They can be monovalent for each antigen, but also multivalent. They're available in various formats. And many of these molecules are in development, reflecting about 20% of the therapeutic antibodies in clinical trials. Now, looking at the short history of bispecifics, the concept of bispecifics has been around for quite some time, already since the early 60s of the last previous century. It has taken some time, however, to develop bispecific antibodies, and this was mainly due to difficulties both in finding the antibodies with the right activity, but also in finding ways to manufacture these molecules. Well, several solutions have been found, and the first products have now arrived, starting with Blincyto, a T-cell engager, a bispecific single-chain Fv antibody that was approved in 2014 in the US and 2015 in EU. And then two further antibodies have been approved, imicizumab and more recently amivantamab, a bispecific EGFR c-Met antibody, a duobody. In development of that, I was also involved. Move on to these two bispecific antibodies that engage T cells, so-called T cell engagers. These are bispecific antibodies that can cross-link T cells to a tumor cell, thereby activate the T cell to kill that tumor cell. There's been a huge interest in these types of bispecific antibodies, but the interest really has not materialized to its full extent. So I still call these T-cell engagers a promise to be realized. Clearly, there have been very high expectations for this particular format. To date, two bispecific T-cell engagers have been approved. This was Removab, cartumaximab, which targeted EpCAM and which was later withdrawn for commercial reasons. And then the second was Blincyto or blinatumomab, a CD3-based T-cell engager targeting CD19. This molecule has really revolutionized the field and generated a lot of interest in the development of these types of bispecific antibodies. And this is also shown by the more than 60 bispecific T-cell engagers currently in clinical development. Now, as said, the majority of these T-cell engagers are bispecific antibodies that engage T cells via the CD3 molecule. So CD3 is the signal transduction molecule of the T cell receptor and is basically expressed on all T cells. Now, novel solutions often also lead to novel problems, and we have seen this in this field. And what we observed is that the therapeutic window of these CD3 based bispecific T-cell engagers is often quite limited. So it's driven by unfavorable toxicity profiles, by cytokine release syndrome, which is dose limiting, and can be severely toxic. We have seen on-target off-tumor-related toxicities, so where the T cells attack normal cells in the presence of the bispecific. We also have seen fairly sporadic efficacy in solid tumor indications. And one of the reasons for this, we believe, is that these CD3-based T cell engagers also stimulate immunosuppressive T cells. So, since they bind to CD3 on all T cells, both effector T cells as well as regulatory T cells are activated, and the activation of these regulatory T cells can dampen activity. And particularly in solid tumors, we think this can be a limitation of this technology. So we therefore believe that it's time to generate a next generation bispecific T cell engager. And the notion here is that rather than recruit all T cells, or this mixed bag of CD3 positive T cells, we have focused at LAVA to recruit one specific subset of T cells, a subset that is specialized in both sensing cancer cells as well as suppressing cancer cell growth and activity. So these antibodies that we have generated are obligate bispecific agonists, so they only activate these gamma-9 delta T cells upon cross-linking to tumor antigens. And we think this may provide a novel, efficacious, safe route and cost-effective opportunity for the development of innovative cancer treatments. Now, first, let me talk a bit about gamma-9 delta 2 T cells. So gamma delta T cells belong to the first line of defense against cancer. So gamma delta T cells differ from alpha beta T cells in that they are able to recognize non-conventional antigens. And of specific interest are this group that we call the gamma-9 delta 2 T cell population there on the left bottom. So these are T cells that via their T cell receptor are able to sense the presence of certain metabolites in cells. These metabolites, called phosphoantigens, are typically present at higher levels in tumor cells. And the presence of these phosphoantigens, and I'll explain that in more detail later, trigger a conformational change in a molecule called butyrophilin, and this is sensed by the gamma-9 delta 2 T cell receptor. And this is the way by which these gamma-9 delta T cells naturally are able to see tumor cells. There's some other additional molecules involved, but this is an important notion to understand. Now, these gamma-9 delta 2 T cells therefore belong to the first line of defense against cancer. They have the potential to elicit potent, direct killing response, but we also think that they would be able to induce more durable responses. And the reason for this is that these gamma-9 delta T cells can orchestrate immune responses. They can activate additional immune cells, and also they have an ability to present antigen to conventional T cells. So these gamma-9 delta T cells indeed are able to bridge sort of this innate immunity with the adaptive immune response. And therefore we think they are very high interest. So in summary, V gamma-9 V delta 2 T cells have an important immunosurveillance function in our natural immunity. They are very sizable pool of T cells. 1 to 10% of all peripheral blood T cells are gamma-9 delta T cells. So from the clonal perspective, it's a very sizable and large population of cells. They have a natural ability to recognize and kill tumor cells, as explained by this ability to recognize both the metabolites as well as additional stress ligands on altered cells. Interestingly, they are also a very homogeneous and highly cytotoxic effector T cell population. So they exclusively are composed of effector cells. There are no regulatory T cells in this population. They have been shown to infiltrate tumors independent of mutational load, and I'll show you a slide on that later. And as we said, they are able to bridge this innate and adaptive immune response, but can both have a direct cytotoxic effect, but also can orchestrate downstream immune responses. For example, due to their ability to present antigen to conventional T cells. Now, the interest in gamma 9 delta T cells was raised by these two important publications in the field. So on the left, we see a publication by Gentis et al. In Nature Medicine, where they studied the presence of various leukocyte subsites in cancer and correlated this presence with response. And interestingly, when one does that analysis, gamma 9 delta T cells came out on top. So it's the single leukocyte subset that most strongly correlate, or the presence thereof mostly strongly correlated with survival in cancer patients. And this is across a wide range of tumor types. Now on the right, Tosselini et al. Followed up on this publication, and they indeed demonstrated that there was an abundance of tumor-infiltrating gamma 9 delta T cells in a wide range of tumors, as shown here, both hematological as well as solid tumor indications. And again, they indeed demonstrated that it is the gamma 9 delta T cells, the presence thereof correlates with, the presence thereof in tumors correlates with survival or response in cancer patients. Now, there's further clinical evidence that gamma 9 delta T cell based immunotherapy has potential. And this is, for example, shown by historic studies where either gamma 9 delta T cells were taken from patients, expanded, and given back in adoptive cell transfer therapies, or in other studies where these gamma 9 delta T cells were activated in vivo, for example by use of the small molecule amino bisphosphonate, which leads to an increase in these so-called phosphoantigens, its metabolites, that leads to activation of the patient's gamma 9 delta T cell in a systemic manner. What these studies have shown is that there is some benefit of these types of approaches. And the second good news was that these approaches are generally safe. So the activation or the systemic activation of gamma 9 delta T cells in patients does not lead to severe safety concerns. There's no signs of cytokine release observed, and this approach therefore is considered to be should be considered to be developable. Now, one observation was the sort of disappointing clinical activity, and we think this is due to the fact that these initial approaches led to a systemic activation of gamma 9 delta T cells. So there's no targeting arm. And we think we can improve on this by using bispecific antibodies. So by using bispecifics, we believe we might be able to activate these gamma 9 delta T cells in the presence of the tumor, and therefore lead to develop a targeted therapy that could be much more effective than these systemic approaches. Now, at Therapeutics, we are developing the gamma body platform. So this slide first summarizes on the left the natural way in which gamma 9 delta T cells see tumor cells. So phosphoantigens accumulate in tumor cells. It leads to a conformational change in these butyrophilic molecules, and this is sensed by the gamma 9 delta 2 T cell receptor. This leads to activation of the T cells and killing of the tumor cells. At Therapeutics, we're now developing high affinity bispecific antibodies that are able to cross-link the delta 2 arm of the gamma 9 delta T cell receptor to various tumor antigens. This cross-linking then leads to a conditional activation of the gamma 9 delta T cells upon bridging with the tumor cell. Our platform is fully modular. These are bispecific antibodies. We can combine the gamma-9 delta T cell binding arm with existing antibodies, with novel antibodies, and this leads to a very straightforward lead discovery and development process which is compatible with all existing antibody engineering standards. And therefore we think we will, with this approach, we will be able to develop off-the-shelf therapeutics which have the advantages of the streamlined manufacturing and well-established antibody products characteristics of this platform. Now first we generated gamma-9 delta T cell specific single-chain domain antibodies by immunizing llamas and then using phage display to generate specific binders against gamma-9 delta T cells. From this we selected agonistic antibodies as shown on the top right. So these domain antibodies can bind and activate primary human gamma-9 delta T cells, of course only upon cross-linking, so the activation is conditional. We also showed that our antibodies bind and activated common delta 2 CDR sequence variants, so there are various sequence variants of the delta 2 gene have been described, and basically our antibodies bind all these common variants. So therefore we selected antibodies that could be used, could virtually be used in any patient. Now let me now talk about EGFR as a target for gamma delta T cell engagers. Now EGFR of course is a very well known tumor target, widely expressed on various tumors. Its overexpression, aberrant signaling, correlates with poor prognosis. And several monoclonal antibodies, canonical antibodies, including cetuximab and panitumumab, have been approved for the treatment of metastatic colorectal cancer and head and neck cancer. This slide shows the binding characteristics and activity in inhibition of signaling of our bispecific gamma delta T cell engager construct. On the left, we show binding of our domain antibody against delta 2 to gamma-9 delta T cells within the parent KD of about half nanomolar, so high affinity binding. On the right, we show binding of our EGFR domain antibody that binds EGF receptor on various cell lines with a KD of around 5 nanomolar. So here we have the EGFR gamma-9 delta 2 cell engager bispecific antibody, and we show that in this case A421 cells are killed potently, very potently, in the presence of the engager and gamma-9 delta T cells with an EC50 of around 4 picomolar. And low picomolar range, so this very high potency is quite usual for the various T cell engagers that we developed with this technology. In the middle graph, we show that killing of EGFR positive tumor cells is independent of downstream mutations. So indeed, this antibody could be used for EGFR expressing tumors with various mutations, including those targeting of activating RAS or BRAF. As I said, an interesting aspect of gamma delta T cell engagers is that they do not activate regulatory T cells. And this is important because for CD3 based T cell engagers, so these are, as I explained earlier, these are engagers that activate all T cells, so alpha beta T cells, as well as gamma-9 delta T cells, as well as regulatory T cells. So these T cell engagers have been shown to dampen responses under conditions where high numbers of Tregs are present. And for example, this has been shown for blinatumomab at a certain cutoff, at a certain level of Tregs, no responses or lack of responses are observed. So this also has clinical relevance. Now, gamma-9 delta T cell population that we target does not contain any regulatory T cells, and therefore they're also not activating. So this is also demonstrated on the graph here on the right, where we show that where we compare a gamma-9 delta T-cell engager and a CD3-based T-cell engager, both targeting EGF receptor. So in this case, SW480 EGFR positive tumor cells were incubated either with the gamma-9 delta T-cell engager or the CD3-based T-cell engager. They are on the second and third line of the legend. And then we added gamma-9 delta T cells, and we added, in some cases, we also added T regulatory cells. And what is observed indeed is that CD107A, so degranulation of the T cells, is induced by the gamma-9 delta T cell engager. If we add regulatory cells, this engagement remains at the same level, so that there is no inhibition of activation. However, if we add regulatory T cells to the condition where we activate the T cells with the CD3-based T cell engager, we see a sharp drop in activity in the presence of T regulatory cells. So indeed, the activation of T regulatory cells by these CD3 engagers dampen responses. We do not also observe this with the gamma-9 delta T cell engager, and we think this may be a benefit in terms of potential efficacy in the clinic, as well as a decrease in heterogeneity in responses, but specifically in those patients that have high levels of Tregs in their tumor microenvironment. Now next, we also showed that our gamma-9 T-cell engagers targeting EGF receptor can activate the patient's own gamma-9 delta T cells. So that's shown on the left and also on the middle. Now next, what I explained before is that gamma-9 delta T cells are able to orchestrate immune responses. And we studied that, for example, by adding tumor cells to peripheral blood mononuclear cells, again in an autologous setting, and then looked both at the activation of gamma-9 delta T cells as well as additional immune cells in these types of cultures. Now the first thing that happens is that the engager activates gamma-9 delta T cells. So we require this activation because if we deplete the gamma-9 delta T cells, no activation occurs. But upon this activation, we see that not only the gamma-9 delta T cells are activated, but during the course of the experiment, and this was a 7-day experiment, we see that additionally also conventional T cells, CD4, CD8 positive T cells, as well as NK cells become activated. So we believe this spreading of the response can lead to additional effector cells being activated and induced to kill the tumor cells. One very important aspect of our technology is that we retain this unique ability of gamma-9 delta T cells to sense the difference between normal cells and tumor cells. And that's demonstrated in this slide. So again, from surgery, we obtained tumor tissues from patients, and which we then divided in tumor tissue and also in some normal tissue obtained from these samples. We then again dissociate the cells and looked at lysis of these cells, of these tumor cells, induced by gamma-9 delta T cells in the presence of our engager, each of our targeting engager. Now, as you can see here on the left, we show potent tumor cell kill by gamma-9 delta T cells in the presence of an engager, shown in red, both for a number of different cancers, including colorectal cancer, head and neck cancer, and lung cancer. Now, interestingly, if we take non-tumor tissue from these same patients, we see that these tissues also expressing EGFR are not lysed. So there's tumor preferential killing of the tumor tissue by gamma-9 delta T cells in the presence of an engager. And we have many such examples. So here we have a clear differentiation from CD3 based engagers, which cannot make this difference. Now, indeed, if these gamma 9 delta T cells are able to distinguish the difference between normal cells and tumor cells in the presence of our bispecific T cell engager, and normal cells are spared from killing, then this would also mean that in non-human primates studies, a benign safety profile would be expected. So indeed, we performed these studies using a number of bispecific gamma delta T cell engagers targeting various antigens, and what I'm showing here is an engager targeting the tumor-associated antigen CD1D, and another one EGF targeting EGF receptor. We generated surrogate molecules that were fully cross-reactive and that were able to bind, activate non-human primate gamma 9 delta T cells against these, in the presence of these tumor targets, similarly to human gamma 9 delta T cells. So indeed, fully cross-reactive system that can be used to study the safety of these types of molecules. Now, interestingly, we were able to dose our engagers at very high doses. The examples I've shown here is dosing up to 10 milligram per kilogram several times during the course of 1 week for our infusion, 4 doses during 7 days of treatment. What we observed were virtually no signs, clinical signs of toxicity. We did see cytokines induced, but the cytokine spikes were very low and did not lead to cytokine release syndrome. And interestingly, we did not see any clinical chemistry abnormalities or histopathological abnormalities. Now, as noted before, this compares very well to studies that have been done previously, and for example published by Lutterbus et al., study by Amgen, reported in PNAS of 2010, where they showed that a CD3 based T cell engager targeting EGF receptor was highly toxic, and a dose of only 31 microgram per kilogram continuous infusion led to severe toxicity in which the animals had to be terminated 2 days into the study. And pathology was observed in tissues that expressed EGF receptor. So this clearly shows that since we were able to dose our molecules several orders of magnitudes higher without any clinical signs or histopathological abnormalities, this we believe supports the very benign safety profile of our gamma delta T cell engager, and support this notion that in the presence of our engager, the gamma 9 delta T cells retain their ability to sense the difference between a normal cell and a tumor cell. So this is where we are. So currently we have one program in the clinic. The second one is moving to the clinic imminently. The first one is Lab 51, is a CD1D targeted gamma delta T cell engager with initial focus on the treatment of CLL, multiple myeloma, and AML. And our second program will be a gamma delta T cell engager targeting PSMA. This is Lab 1207. As I said, the clinical trial application has been approved, and this molecule is moving to the clinic at any time now. Third program I talked about is the Lab 1223. Is a program targeting EGF receptor, and I showed you the data for that study. We showed high potency against EGFR positive tumors, sparing of normal cells, and a very benign safety profile in non-human primates. This molecule is moving forward, and we're expecting an IND end of next year. Thank you very much for your attention, and goodbye.